So first of all, I will start with Environmental Engineering 1 course. So in this, we'll discuss some environmental factors. First of all, that how environmental engineering is linked to the transportation engineering. They both are interlinked. Firstly, the objective of this laboratory session is to determine the residual chlorine in water sample by phenyldiamine DPD in short method using spectrophotometer, in which we have these two reactions that is interchangeable. The sum of HOCl and OCl is called free residual chlorine. What is the practical significance in this lab is that their determination is used the assessment and control of receiving water quality and the treatment and supply of potable water. It's like that how we can clean the water by using certain methods and using certain chemicals. So here we'll discuss certain things in detail. First, on the other hand, we have chlorine utilization in water, how the chlorine absorbs water, and how the water is treated using chlorine treatment. So chlorine is used by organic matter and impurities. Chlorine demand and remaining chlorine is total chlorine. So first we'll discuss what is total chlorine. Total chlorine is basically used by nitrogenous compound and not available for disinfection, that is combined chlorine. And on the other hand, we have chlorine not used in any reaction and available for disinfection in water, that is free chlorine. Then what is the requirement for this lab test? First, we need the list of equipment, that is chlorine test kit, that is DPD, and here we have the kit. And then we need test tubes for the water which we are treating. We'll use the water in those test tubes. Then we'll use spectrophotometer. And what are the reagents that will react with water? First reagent is chlorine, and secondly, mm, here we have reagents. Now let's come to the procedure. First, we'll describe about the determination of free chlorine. So the procedure starts with fill the sample. Here sample means water sample into the 10 ml cell of spectrometer. Add 1 chlorine reagent and shake it vigorously until the reagent is completely dissolved. Leave to stand for 3 minutes. That will be our reaction time. Enter the stored program number for free chlorine and total chlorine at 530 nanometer. NM here means nanometer. Place the distilled water sample into the cell holder. Distilled water is the tap water. Insert the 10 ml cell riser into the sample compartment and press 0. The display will showing 0 on the machine that we are using to determine the test. Now insert the sample in which reagent chlorine is already added and note down the concentration of free chlorine in mgl, that is milligrams per liter. Second procedure we have is immediately add reagent chlorine and mix it. Leave to the stand for 3 minutes. Fill the sample into the 10 ml sample cell of a spectrometer. Enter the stored program number for total chlorine at 530 nanometer. Now insert the sample in which chlorine is already added and note the concentration of total chlorine. Combined residual chlorine that will also be measured in milligram per liter. Total chlorine minus free chlorine. Uh, now now here we have laboratory session 8. That's objective is to determine the presence of chloroform in water sample by most probable number, that is MPN method. The previous method was DBN method, DPD method, and now we have the MPN method. Here we have the description of chloroform and its practical significance and the equipment used and the reagents and the procedures. So, firstly, the equipment. Let's first start from the chloroforms. What are chloroforms? Chloroforms bacteria live nearly in the human intestinal tract, and an average person will excretes billions of them in a single day. Chloroforms bacteria are non-pathogenic, but their presence in water is as indication of fecal contamination. They are indicator organisms, and their presence presumes the contamination has occurred. Its practical significance is that the chloroform group of bacteria is the principal indicator for suitability of water for domestic, industrial, or other uses. This method indicates the degree of contamination. Now we have the equipment required, that is fermentation tubes, that is test tubes, Durham tubes, autoclave incubator, platinum loop, burner, pipette, test tube, wrap, and the reagents here we using are lactose broth. So the procedure we have is, again we have two procedures. First procedure is set up a series of 15 tubes of a lactose broth in the test tube tract. Label as to the sample and volume of a sample included using a 10 ml sterile pipe. Transfer 10 ml of sample to 5 tubes labeled LB2X 10 ml using 1 ml sterile pipette. Transfer 1.0 ml of water sample to the 5 tubes labeled. Lastly, add 0.1 ml of sample with the help of sterile pipette to 5 tubes label. Incubate all the tubes at 35 degree, 37 degree for 24 to 48 hours in an incubator. Second day session is examine all tubes after 24 to 48 hours of incubation. Formation of gas occupying at least 10% of the Durham tube is indicate if a positive test for the presence of chloroforms. When there is no gas in the tube 48 hours, the test is negative. That means that chloroform isn't present. Here we have the pictorial representation of how the procedure will occur, and here we have the values that will occur in during the um, fermentation and incubation of the process. Here we have. Now for this we will end here and we'll continue the next lab test inshallah in the next video.